Again, this is actually the nomenclature of nucleic acid that basically gives the different nucleotide, and nucleic acid of the different nitrogenous bases. As I've said, the difference between purines and pyrimidines is that the nitrogenous bases that includes in purines are adenine and guanine, while in pyrimidines, it actually have a cytosine, thymine, and uracil. So the difference between the nitrogenous bases of the DNA and RNA is that DNA has adenine, guanine, cytosine, and thymine, while RNA has adenine, guanine, cytosine, and the thymine will be replacing with the word uracil. So always remember that one. So as you are now asking me what are the difference between nucleoside and nucleotide. Okay, I know you are questioning yourself in what is the difference of it. Basically, nucleoside is bottom, or it is made of nitrogenous bases, usually either a purine or a pyrimidine, and it has a 5-carbon carbohydrate ribose. But nucleotide is simply a nucleoside with an additional phosphate group or groups of polynucleotides containing the carbohydrate ribose are known as a ribonucleotide or RNA. Okay, as I've said, nucleotide is simply nucleoside. The difference of it is that nucleotide is a nucleoside with an additional phosphate group, while nucleoside is actually referring only in the purine and pyrimidine. But basically, the difference of it is nucleotide has an additional phosphate group or a group of polynucleotide. Okay? So, as you can see, we have some, literally, the purines in adenine, it makes adenosine for the nucleoside and adenylate for the nucleotide. And of course, in the nucleic acid, we have RNA. So, that is actually the summarization of the nomenclature of a nucleic acid. If the base has a nucleoside, or the base has a nucleotide, or the base in the nucleic acid. So always remember that one. Next slide. So what can you observe with this ladder? So it's actually defining or showing the structure of the secondary DNA, or it is actually shows the secondary structure of the DNA in a double helix. So as you can see, the ladder spiral, that is actually the structure or the illustration of the DNA. Next. So as I've said, DNA, the secondary structure of DNA, we have two antiparallel, like this one, two, and this ladder, this line, this line and this line. So we have two antiparallel polynucleotide chains, one around the same axis. We have sugar phosphate chain wrapped around the periphery, and we have the bases. We have adenine, thymine, cytosine, and guanine occupy the core, forming complementary. Okay, so this is actually the structure of the DNA. Next, so let's go now on the rules in pairing the nitrogenous bases. So it calls as a Chargaff rules. Basically, the Chargaff rules compose of 4 rules. We have 4 steps of rules. We have base. The pairing of the base rule is the adenine and thymine, and guanine and cytosine. So in the DNA, but in the RNA, thymine is replaced by uracil. So the bases are bonded to each other by hydrogen bonds, of course. It discovered because of the relative percent of each base. Notice that the adenine is bonded into the thymine, similar to the other nitrogenous bases. We have cytosine bonded to the guanine. Okay, next slide. So this is basically the function of DNA. DNA is used to store genetic information. As I've said, aside from storing, DNA also carries genetic information or genetic material. It by the process of it is actually replicated before the cell division. So DNA is very important in a storing of the nucleus. It never leaves the nucleus, yes, and your DNA stores the code for your protein which exhibit your traits. So in that process is called in the protein synthesis, and the DNA gets converted to RNA in order to move out into the cytoplasm. That is actually the reason why we have transcription and translation for the RNA to be made of. Okay, to be the components of RNA, or how RNA made of, because of the transcription and translation. That is why DNA gets converted to the RNA in order to move out the cytoplasm and to have the process in the protein synthesis. Okay, next slide. In the cytoplasm, it meets up with the ribosome where it can synthesize proteins. Of course, by synthesizing it, the DNA now will store genetic information. Aside from storing, it will maintain the growth and repair, controls all the cellular activities, contains protein codes, and of course, ensure each daughter cell and gametes receive exact genetic information. Okay, let's now unlock some difficulties here. Let's define what is gametes. Okay, let's now define what is gametes. Okay, basically gametes is a mature haploid male or a female germ cell which is able to unite with another of the opposite sex in the sexual reproduction forms for zygote. So what exactly is a gamete? Okay, basically gamete is a sex or reproductive cell containing only one set of dissimilar chromosomes or half of the genetic material. So that is why in the function of DNA, it ensures that each daughter cell or a gamete receive exact genetic information or genetic material. Okay, why? Because it is necessary to form a complete organism. Okay, continue. For the DNA, so this is the summarization of what is DNA and what is the components of the DNA. It said that the abbreviation of DNA is deoxyribonucleic acid. As I've said, the sugar of DNA is deoxyribose. It is a polymer of deoxyribonucleotide. When we say polymer, okay, when we say polymer, again, when we say polymer, it is a substance or material consisting of a very large molecules or macromolecules. Okay, DNA also is a thread, like a structure is a combination of a large number of nucleotide units joined together. So as I've said, in a nucleic acid, it is composed of polynucleotides. When we say polynucleotides, a large number of nucleotides bonded to another nucleotide and bonded to another nucleotide. So that makes it polynucleotide. So this polynucleotide again contains genetic information or genetic materials that gives rise to chemical and physical property of the organism. So that makes the insurance of the daughter cell and the gamete with that receive exact genetic information, and it is necessary to form a complete organism. So that's the reason behind that one. Okay, next slide. So let's go now to the location and the isolation of the DNA. So for the location of DNA, it can be found in the chromosome, specifically in the nucleus, of course, and in the mitochondria and chloroplast of the cell. So in the prokaryotic and eukaryotic cell, it can be found in the mitochondria and chloroplast. But basically, in an organism, it can be found in the chromosome, specifically in the center of a cell, which is called nucleus. So in the isolation, it is present also in every living organism because it contains genetic material. So for the isolation from viruses, bacteria, thymus gland, spleen, blood, hair, skin, and etc., DNA is actually basically there. So in an isolation process from the viruses, bacteria, and thymus gland, spleen, blood, hair, and skin, and etc., DNA is there. Okay. So basically, there are steps in the isolation of the DNA. So please take note with this. The 3 steps of DNA extraction are lysis, precipitation, and purification. So I will be digging deep in that properties in our discussion in the structure of the DNA. So but now let's move to another slide. Okay. So let's go now to the size of the DNA. Okay, size of the DNA shows a great variation. And basically, size of a DNA is from the 1.7 angstrom wide, long in simple structure of a simian virus with a 5 or 6 genes, and can also extend to 2 meter in a human DNA. Okay, so always remember that one, that in the size of DNA is only 1.7 angstrom wide, long in a simple structure of simian virus with a 5 or 6 genes, and can also extend to 2 meter in human DNA. So the size of a human DNA inside the chromosome is just a true 200